Thank you.
Yeah, we wanted to thank you very much for being part of the translational series here. We want to also say that you and your team are hosting the Foundations of Biobehavioral Health Symposium, right?
Yes.
Which is what, Thursday, May 18th?
Next Thursday.
And so we typically have what we call our translational topics at noon. We'd encourage everyone to instead register for the symposium and go and see lots of interesting work that's going on there.
Yeah.
So
you don't have to come to my keynote because it'll be a little similar to today.
Well, that's okay. That's all right. There's information out.
It will be a little different.
Yeah, sure. And there is information out both on our website as well as through Health and Human Development listserv, depending on who you're connected to, about that event and how to register.
Fantastic.
All right. Welcome, and we'll turn it over to you.
Okay, thanks. Hi. So it's going to be awkward because I'm going to like this. And then I know there are people there on that blank screen that I can't see, but you can see me. So we'll just do the best we can here. So I wanted to talk today about what we now in the field call precision-based modeling. And there's a great deal that we think this is really where the field of prevention science is going. Oh, there's another slide here that that I think Sarah put in. Sarah?
Well, that's true.
I'm sorry. So do you want to talk about that first?
I will. This is just an announcement for another event. Thank you. Sorry, I forgot that was
I forgot it was
our informatics core is holding a session, sort of a retreat to talk about exploring these challenges of integrating analysis across all kinds of medical data. And that's next on the 19th, which next Friday, I believe, 1 to 4 over at Knoll Lab. And it's an important conversation across the university, so we'd encourage you, if you're interested, you can register and come to that as well. We'd like that to be a broad conversation.
Okay. Sorry to be taking notes.
No, that's okay. And again, this went out on the HHD listserv as well as OVPR, so depending on how you're connected.
Okay. All right.
Great. Thank you.
Okay. All right, so now here's the original slide that starts the presentation. And so there are a number of reasons why the field is moving in the direction of being more precision-based. Certainly psychiatry and medicine and so forth are already going towards a personalized medicine approach. And what can we do in prevention? You know, usually preventive interventions, not always, but usually are universal. And so you're reaching a lot of people. You don't know exactly who's catching, you know, what you're throwing at them and who's going to be responsive and who isn't. So what we are trying to do in this program of research is really characterize people, really do person-centered analysis to find out who's responding and why some are not responding and so forth. And so we're looking at really underlying mechanisms now in responsivity. And so we don't run like just regular RCTs to determine the efficacy or the effectiveness of an intervention. Instead, we take interventions that have already been determined to be evidence-based and have, decent effect sizes. And I'll get into the effect size issue in a few minutes. But we want to get something we already know is going to affect at least some of the population so that we can further characterize the individuals who are receiving the intervention and determine who's responding, who's not. So when I first started this program of research, this particular one, I guess was about 15 years ago, if I recall. I'm not really that old, but I think it was about 15 years ago when I started really looking at interventions. And it was very difficult to get through review boards at NIH because they were like, well, it's supposed to be an RCT, but you don't have enough schools, and how are you going to deal with nesting and all these other issues that are really specific to trying to determine the efficacy of an intervention. That was not our purpose at all. With the first study that we did of this nature, it took 6 submissions. Mark Greenberg was my co-PI. He was the former director of the center that I have now. And it took 6 submissions, and you're going, well, how can that be? You can't have that many submissions to NIH. But back then you had 3 submissions, and then if you didn't make it, then you had to completely reframe it. And so what we did was we changed the title because we felt really strongly that this was the way to go. So we changed the title of the proposal, and we did enough in the specific aims to reframe it enough so that the CSR, who determines where it goes, couldn't tell that it was the same submission. And we finally, at the sixth time, got it and have finished the study. And I will mention it later, but just to say how difficult it has been in prevention science for reviewers and the field in general to begin to adopt this perspective. And now there are a number of people, not many, but enough to start to look at the underlying mechanisms and trying to be more precision based. And so this talk focuses on neuroscience. So it doesn't have to be neuroscience to look at underlying mechanisms. They might be social mechanisms. They could be behavioral mechanisms. But we're interested in neurodevelopment and ways in which neurodevelopment can impede the ability of an individual to respond. I should look forward to of an individual to respond to an intervention, or if neurodevelopment is going along a normal trajectory, then it would explain why individuals are maybe able to do better in some of the interventions that we offer them. So we've done in identifying where to look, we have done previously a lot of etiological work. So a lot of this depends on the etiology of the phenomena that you want to prevent. So you're trying to be precision-based with respect to targeting that etiology so that you can make, alter that trajectory and therefore, start to make behavioral improvements. So we did a lot of longitudinal, so we're still doing longitudinal studies with respect to drug abuse. We have looked at the precursors to drug abuse prior to children using, and then looked at the consequences of the eventual drug use. And this helps us to understand what the predictors of drug use are and a lot of other risk behaviors, right? Because we have to, look at multiple domains. We do hours and hours of testing and assessments and so forth. And then we have a better idea of what it is that we need to target. So in a translational neuroscience prevention program of research, our objectives ultimately are to determine what works best for whom, why, and under what circumstances. So what is the intervention? What works best for whom is moderation. What are the characteristics at baseline that predict a favorable or unfavorable response to any given intervention? And then why? Why is mediation? What are the mechanisms? I personally am not interested in statistical mediators. I want malleable mediators that are statistically significant, but you can do statistical models and find mediation with mechanisms that are immovable. Who cares? For the field of prevention, we want to change these conditions so that we can lead to improvement. So I'm not interested in certain mediators that can't be moved. Of course, we are interested in ways in which sex differences can portend different outcomes in intervention. You can't change, well, you can change sex, but it's not that malleable. And race and environmental conditions and poverty. There's certain things you can only move a little bit, but it's good to know that these are moderators so that you can tailor your interventions to match those characteristics, knowing that they're going to respond differentially based on them. And then the why, again, so the mediators that we tend to look at in this program of research are neurodevelopmental, but we also look at some of these other characteristics in interaction, like sex and race. And then under what circumstances? So what are the contextual and experiential factors that determine who a person is and how they're going to respond and orient themselves to different environments. So what are those circumstances? But in other cases, circumstances can be those conditions under which an intervention is implemented. So it may have to do with the process of implementation. It may have to do with the fidelity. in a lot of our intervention, in all my studies, they're in high poverty neighborhoods. And so I can't begin to tell you how difficult it is in some schools where they don't even have, outlets that work, and you're trying to do stress physiology and you've got nothing to plug your equipment into because the outlets are so poorly connected, and they're screaming in the hallways. And, a lot of these studies are in Baltimore City, and the circumstances under which the interventions are implemented are quite difficult and almost impossible to control. So we have to take some of that into consideration and actually do measures of school climate and so forth. So as I said, we want to apply an etiological understanding of the behavior that we want to prevent to our intervention research models. So what do we know about the underlying causes? In the field of prevention, and I don't know if is anybody here who does prevention work? Yeah. So what is known but not widely acknowledged is that even the best interventions only have small to modest effect sizes. So our best evidence-based interventions only reach subgroups of people. And so what I've been interested in actually since I was in 5th grade is why are only, how are these subgroups responding differently? What are the underlying mechanisms in their responses? How do we reach those? How do we reach individuals that are not responding well? Because think about what the implications of having small to modest effect sizes are. It means that a lot of people are not responding, but they're statistically significant. And yes, you can publish all day long, but the clinical utility is not optimal, right? So we want to be able to understand the characteristics of differential responding. We do not know to this day what the characteristics of favorable responders are to conventional treatments. We don't even know why some things work for some people. And not a lot of people are looking at those kinds of mechanisms. And then we need to try to identify the characteristics of the heterogeneous subgroups that are unresponsive. They're not just one big amalgam of individuals that all look alike. They're all not responding for various individual level reasons that we need to elucidate. So we want to understand these mechanisms that explain intervention outcomes, and then determine how the interventions modify these underlying processes. So this is our cumulative model of risk for psychopathology. It's really consistent with the literature, but Emma Rose, who I brought with me we were at University of Maryland School of Medicine prior to this, so she's a neuroscientist, and developed this depiction that I think is really nice because it makes it nice and easy to understand. But in this model, we have basically two sets of influences. We have the genetic risk variance, and again, this is part of the etiology that's important to understand the behavior that we're trying to prevent. So this is how this is relevant. So we have the genetic risk factors that are either turned on or off, right? They're largely predetermined. And then we have interacting with those genetic variants are the environmental gauges. These are like dials that are ramped up or down depending on what the individual is exposed to. Some of them are adverse conditions. Some of them are, will promote resiliency. And so you see the red versus the green. And the red is, you probably can't read it on yours either. I can hardly read it on mine, but it's like discrimination and poverty and child maltreatment and family history of mental illness and drug use and so forth. These are all, and there are many, many more, adversity, and everybody's dialed up or down, and these dials go up and down throughout life. And so there's a developmental trajectory that has to be considered in prevention too, because they're not static here. And they also, very dynamically interact with these these genetic risk factors. Together, they act to determine the extent to which the brain structure and function have integrity. So they directly affect brain structure and function. And then to make it more complicated, we now know that epigenetic modifications are very important in this process, and that you probably can't even see it, but that arrow really should be going in the other direction. I couldn't fix that last night. It was too late at night for me to start messing, and y'all needed my slides. But anyway, so epigenetic modifications further determine how genes are going to be expressed or suppressed. And that in turn, has a lot to do with what the phenotypes are. These are the intermediate phenotypes. And again, only a few examples here that I can hardly read, but it's like novelty seeking and impulsivity and cognition and behavior of various kinds, aggressiveness. So these are your intermediate phenotypes that are expressed by the brain that are a function of these interactions. And that together determines your place on what we call the liability threshold. So it's, and it goes up and down throughout life, and it can be changed. And so the whole idea of prevention is to bring the threshold down, right? And what prevention primarily does is focus on these environmental gauges to ramp the resiliency gauges up and ramp the adversity gauges down. So these are some of the etiological considerations in our prevention protocol. So we need to consider the genotype by phenotype by intervention effects and their role in outcomes. We do believe, we have every reason to believe that intervention, if it works well, for people in whom it works well, has potential to enhance neural growth and development and may have an enduring effect on the behavioral trajectory. If you think about it, it's we hear about, the term for better or for worse. The worst part of the equation is all the red stuff and that other, where we know very definitively that if you're exposed to child maltreatment, if you live in an impoverished environment, etc., the literature is replete with studies that show that it has a very detrimental effect on the development of the brain. And that is something that we know very well. So if these external influences can influence the way the brain develops, why can't positive influences be the for better part of that equation? For better or for worse, the brain is experience dependent. And so we want to capitalize on that knowledge. And we think that if we could get these targeted, specific, sometimes novel intervention components, and we can match them to well-defined needs of subgroups who are otherwise not responding to conventional treatments, then we have a more precision-based approach. So early intervention is desirable. The brain is most plastic and susceptible to lasting changes before the problems become entrenched. I will tell you, though, that you can change the adult brain. If you can change the adult brain, you can change the child's brain much more easily. The child's brain is much more plastic. But you can change the adult brain. CBT, cognitive behavioral therapy, has been shown to change the adult brain within 12 weeks in those that it works for. So again, we have to think about the subtypes at all times. So, and I'll mention that again. So basically, in our model, the brain is a mediator for risk behaviors. So adverse developmental contexts lead to being high on the liability threshold. They affect the brain, they hurt it, and that's what leads to high-risk behavior. So that's just the very basic model. I like to focus on adolescence when I give this presentation because it's such a dynamic period of development. And when I was going to school, which was, again, a few years ago, we were encouraged not to study adolescents because, my gosh, they're so changing, they're so out of control. There's all these hormonal things going on, and there's such huge differences in the developmental trajectory for adolescents, some maturing at 14, some maturing at 10. And so how do you control for all these factors? Well, we've discovered since that adolescence is probably the most interesting part of life, not only personally, but for the researcher, because of all these dynamic changes, and the plasticity is very high in adolescence. I still to this day will have people ask me, people like judges and police officers and legislators, and saying, because we do a lot of outreach, isn't it too late by the time they get to adolescence? Isn't it just too late for them? No, it's a very malleable period, and it's a great time to intervene, but certainly the earlier the better, but we definitely don't want to exclude adolescence. And of course, we know now that adolescence goes on forever. I keep telling my kids that. They're in their 20s. It's like, your brain isn't connected yet. You can't do that. But we're legal adults. Yeah. The legal age of 18 means absolutely nothing in neurobiology. So it's very normative. It's very normative. This is not an abnormal state. The brain is still under construction, particularly with respect to the connectivity between the prefrontal cortex and limbic system structures. So why do most 16-year-olds drive like they're missing part of their brain? Because they are. And they also engage in a lot of risk, and they are, you're more likely to actually die in adolescence than any other time of life, from accidents and injuries and drug use and doing really risky, stupid things. They occur mostly in adolescence. But there's, a neurobiological reason, and there's an interesting evolutionary reason too that I won't go into. We'll do evolution another time, but there's a reason for everything. Okay, so the frontal cortex is the last to develop, as I mentioned, and it really isn't fully developed as far as we know, until individuals normatively are about 30, until they're almost 30 years old. And so they're still establishing their connections and integrating and becoming increasingly efficient over time. And so that's why in the teen years and puberty, the teenage years, adolescence does not really end until you're almost 30. So it is a very protracted process. And I have another evolutionary theory about why it's so protracted, because I really don't think it was protracted in our history 100 years ago, or even 50, 60 years ago, when we had children when we were 15 and 16, and we were responsible and we worked and we had social demands that were consistent with being responsible individuals. And I'm willing to bet you that the brain was much better connected then than it is now in our relatively laissez-faire society. So that's just a little commentary, but it's experience dependent. So if you don't stress the brain, if you don't give it responsibilities and social demands, it may take longer for the brain to fuse. Okay, so it would be interesting to study the Amish and military populations along those lines. So anybody wants to do that, I'm totally game. I'm collecting preliminary data. So anyway, our focal point, and I'm going way too slowly because I'll end up with no time. 4 o'clock. Okay, my watch stopped when?
Yesterday.
Honestly, it's 4 o'clock.
It's about 12:25.
I'm really over. Okay, so our focal point is executive functioning, because that is what is modulated by the prefrontal cortex. And so these are the things that tend to be in deficit when we see behavior that goes beyond what's normative in terms of high risk. So when you have executive deficits and lack of prefrontal control, and I'm going to call this vertical control in a little while, a lack of prefrontal control over these lower centers, these emotional response centers, then you're more likely to have some of these problems with, maladaptive stress responses, negative emotions, impulsivity and novelty seeking, and so forth. So this is what we focus on in terms of neurodevelopment. You may have seen this slide before. It's used a lot. It's a dual process model of adolescent development. And I think it's a really good way of showing what what's going on in adolescence. You have these linear improvements in inhibitory control, largely due to the development of the prefrontal cortex. And just to say that if you test adolescents without peers around, they perform fairly good with respect to prefrontal cortical function, cognitive functions. They do pretty well if you just do cognitive testing on adolescents in a vacuum. But if you put peers in the room or in a virtual simulation, you will have them performing as if they're much less mature. And so that's where you see some of the deficits coming out. So there are very big peer influences. So interestingly, in sort of this contrast is that, you know, the systems that underlie reward sensitivity are really developing much more quickly, and then after adolescence come down again. So there's this inverted U shape that we see where we have a lot of sensitivity in the nucleus accumbens and so forth, the ventral striatum, so that you have a lot more reward sensitivity relative to inhibitory control. So what do you end up with? Basically hedonism. I'm being kind of silly, but you know, you're going to go for things that are rewarding and not pay too much attention to the punishments. So you get behavioral phenotypes of risk-taking, novelty-seeking, and impulsivity, which increases risk for accidents, violence, STDs, and drug and alcohol use. We also have tremendous amount of influence from stress in the environment that acts on systems that modulate brain development and function, so they directly affect executive function, emotion regulation, and reward processing. So, I just wanted to mention that stress plays a huge role, and in our studies, because we're looking at you know, places like Baltimore City and so forth, and Washington, D.C., that they're impoverished. There's a high rate of child maltreatment and witnessing violence. So trauma pervades in these children, which further dysregulates the ability of the prefrontal cortex to control emotional responses. And then I'm just going to mention very quickly, this is where epigenetics comes in, and I'm very excited about the potential role of epigenetics in prevention science research, because it really seems to be at the core of gene by environment interactions. And it's, you know, if you have these, if you have exposures to negative influences, you can actually change the way genetics expresses, the way genes express themselves. But what's really interesting for prevention science is that you can also help to normalize the expression of genes with interventions that are very targeted. And a lot of this work is being done in primates, but there's increasing amount of epigenetic work in humans. And in the primate work, it's showing that if you intervene at one generation, let's say you give a parenting program at one generation, yes, even primates need to parenting, there's a lot of variability there, that it will have an effect in 2 subsequent generations that do not directly receive any intervention. So it's a very exciting time for prevention science, in my view. Now I'm going to talk about where we're at with respect to what we're using for interventions. These are categories of interventions across developmental phases. And the reason why I have this here is just to remind myself to say we have a lot of good interventions. We know enough that we can have an effect, that we can have a beneficial effect on a lot of children. But the problem with all the interventions in each of these categories, as I said before, is that they don't reach everybody, okay? So in my center, we deliver a lot of interventions to the community. What I'd like to bring into the center is the ability to start looking at how these individuals are responding differentially to these interventions that we're delivering. So we have a very much smaller little circle there, oval, for favorable responders. This is the way it usually looks, proportionately. And then amongst the poor responders, different types based on the different things that are preventing a good response. Okay, So, and then we want to look at what the mechanisms of effects are, and we want to look at the experiential and contextual influences and do the, well, I might have that in another slide. Yeah, I have it, I think, in this slide. So the preventive intervention in this model then would act directly on the brain, and those are little fix-it things in the brain, if you can't tell. You saw where the damage was. Now this is tools that are fixing the brain to lead to desirable outcomes. Always have to consider the moderators. And we want to look at these mechanisms, and we want to identify the biomarkers that are indicative of efficacy for various subgroups, okay? So these are some studies that have been done that focus on something physiological or neurobiological. There, you can see just a handful. There's probably 3 or 4 more. That's how many so far in the field of prevention. There aren't very many, but they're really starting to, people are really getting interested, and there are a number of studies underway that will soon be on this. And then some framework reviews. If anybody wants this slide, I can send it to you. These framework reviews are the framework of doing this sort of work. And these 3 articles are especially good for that. Just to say that there are some studies underway. I'm not going to show you the actual data from this, but just to say that this is the study, the PAD study, Promoting Alternative Thinking Strategies. Have you heard of it? Is one of the best evidence-based programs that was developed by my predecessor and his colleagues. And it's, you know, already known to be evidence-based and so forth. I'm not interested in direct effects. I couldn't care less. I'm interested in why some kids respond and some don't. So I did this in Baltimore City in kindergarten through 2nd grade in high poverty neighborhoods and looked at mediation and moderation. So the turtle is the sign of PAS because children are taught. It's a very cool program. I'm really excited about it. We actually got unbelievable direct effects because if you don't have good fidelity, you can't look for mediation. So we were just so careful. We had behavioral effects at 0.01 level in every single behavioral outcome. And the kids learned vertical control. They learn prefrontal control over their emotional responses. And one of the things they do is they turtle. So instead of you know, going off like the way kids do, you know, if they learn to recognize their feelings, and if they're feeling angry or sad or distressed in any way, they will stop and they will turtle. They'll hold themselves as if they're in a shell. And so they're turtle, and so that other kids recognize that they're turtling, and they will come to them. You can see these little kids, these little 5-year-olds, and they come to them and say, you know, what's going on? And the kid will say, I feel mad, and this is my issue, and I'm mad at you, and then they talk. These 5-year-olds, they're not used to talking, they're used to fighting. Literally, they're used to fighting. And then it was interesting because they go home and they turtle at home because their homes are full of conflict and coercion also. And so we were having parents call and say, my kid's doing this, we're turtling. And, you know, and it's like, well, then it's an opportunity to teach the parents to recognize that their child is distressed and how for them to respond with words, soothing words and so forth. So it's a fascinating program. So we found strong and very strong, you know, direct effects. Again, I needed to have that, but it was not of interest in the study. And we found that inhibitory control and emotion regulation moderated the effects. We also found stress physiology. We did stress physiology in these little kids, looked at heart rate variability and skin conductance. And we're finding that those that have both, it's sort of a double-edged thing where you have the kids that are flat emotionally, because when you've experienced a lot of trauma, you go flat, you kind of downregulate, or kids that are having really amplified. So there's either this big amplified response that's moderating or this flat response. We have to tease that out further. But it's causing them to respond less well to the intervention. So in other words, those kids that have less inhibitory control, less control over their emotion, and aberrant physiological responses to stress are not doing as well in the program. So this is really important to note, because maybe the program can now be refined to be more trauma focused for certain populations of kids. That would just be one example of what you might do with these kinds of results in a real world setting. So it may explain why the conduct problems in the subgroup prove refractory in the context of these interventions, because they're so stressed that they can't assimilate the curriculum materials. And what's also true is when they can't assimilate the materials from a preventive intervention, they can't do so from their educational programs either. So they don't do well academically as a result. So I love mindfulness. Well, I can't look at my watch. It's 12:36. Okay. So I love mindfulness studies because they are showing consistent effects. Most of the research is done in adults, young and regular adults, but there's not enough that is being done in almost nothing in children and a little bit in adolescents. But these are studies that use brain indices of how well they're connected and how well they're functioning neurobiologically. And they're finding generally very significant effects of mindfulness on these neurobiological measures. So this is just a sampling. With the other slide, I had almost the universe. With this one, I just have a sampling because there are so many. And there's some really seminal work being done by the mindful compassion speaker that we had a few months ago in the Prevention Research Center, Richie Davidson in Wisconsin, who's been doing this work for about 15 years, well, maybe 13 years, that is showing change in the brain with mindfulness interventions. And so we're seeing some really good effects here. And so what I tend to do with some of these, and I did a study of yoga. I don't know how to do yoga myself, and I'm not a mindful person, but I'm going to be, because I need to learn from this research because it's just fascinating the way in which it helps regulate stress physiology and really help cognitively in multiple domains and so forth. So this is just an example with this one kind of intervention of how you can sort of disaggregate the components of an intervention so that you can determine what the likely mechanisms of effects would be. And that's really important in a prevention program. This sort of research is to map the components of an intervention to what you think are maybe like the neurocognitive prerequisites of responding to that program. And those are the mechanisms that you think will change if the intervention is working. So these are the different components of yoga. I don't know how to pronounce this stuff that's not in English, but you know, they teach us controlled breathing and their postures, and it very much focuses on attention, and there's meditation, self-awareness, and that sort of thing. So you tease all that apart, and then you figure out what it is. And so this isn't a methodological model. This is a conceptual model, but it shows, you know, how we're going to see what it does to the brain if you're trying to, say, retrain attention and self-awareness and mindfulness. It's going to have an effect on the brain. And you're going to see it in executive cognitive function and emotion regulation measures. We use tasks that, you know, performance tests that measure the stuff. And then yoga and mindfulness and so forth increases vagal flow. So we want to look at, you know, this as a physiological substrate of attention and emotion regulation. And then we expect it to have an effect on these outcomes. And so that's one way that we can get our conceptual model together, and we can do this for every intervention. And I think it should be done for every intervention so that we know exactly what's going on and who's not getting it so they can be refined, and we're not just continuing to put interventions out there that are only reaching subgroups. This is from Katie Witzkowitz, who's done really a lot of the seminal work in this area, and she just has the proposed mechanisms of actions and what the processes are that underlie it, and then what exactly is going on in the brain. So I like this slide, and it talks about what's bottom up with respect to emotion, you know, with helping people to regulate their emotion from the bottom up in a way that will help them focus on what's salient from a cognitive perspective, right? But it also talks about what the top-down processes are to help inhibit inappropriate behavior. The good behavior game is another one, so we've modeled for this as well. But anybody know what the good behavior game is? I can't see you all out there, but I know you're there in the blackness. But have you ever heard of the good behavior game, anybody? So it is probably one of the, I shouldn't say probably, it's one of the best evidence-based school programs that's out there for prevention. Has been shown to reduce psychiatric problems later in life. It reduces drug abuse. It improves academic performance. It has a number of really well-established outcomes across multiple populations in different settings. Dennis Embry has a version called PAX GBG, and he's using it literally all over the world. He's doing the entire province of Manitoba, every school in it, which is huge in Canada. I couldn't tell you how big it is, but it's this big on a map. And he's got all these schools participating in this.
Wow.
And there are some schools out west, some counties that have actually incorporated good behavior game into the school curriculum. And it has modest effect sizes, so it's one of the better ones, but still modest. So who's responding, who's not? What are the neurobiological predictors? Let's look at it to guide program refinement so that it can reach more people. It's a very cool program in that it focuses on reward mechanisms. It's entirely about reward, and it's about kids being attentive to their peers. So you don't get rewarded as an individual, you get rewarded in groups. Kids form groups, and if the whole group does well and doesn't have a lot of infractions, and the kids with infractions don't get punished, but the kids that get the reward means that for 3 minutes they can do it, let's say they can do something kid-like that they normally can't do in a classroom. So one of the rewards might be jumping up and down and screaming for 3 minutes. These are the kinds of things that kids love to do, and they're normally inhibited from. So they can, act out for 3 minutes in a controlled way, but something that or they can laugh at uncontrollably, so they get to do fun stuff. But they become very aware of what others are doing. So in the model that I have, it's in review right now. Pray for me. Because I know, it comes back, well, it's not an RCT. Okay. But so breaking down GBG into its components, we figured we'd focus on inhibitory control, emotion regulation, and error monitoring. It's an fMRI study. So we'll be using these tests in a scanner. The kids will be between 8 and 11, so they're not little tiny kids. And it'll be pre-braces, so we don't have to worry about braces. That's always a pain in the butt. We have another study where we have to keep taking people out. So that's what we're focusing on in the scanner, because and error monitoring is what's different from some others, because you really have to be looking at what your peers are doing with respect to your own behavior. And so GBG, fixes the brain to lead to improvements in these neurocognitive functions. And then we have something else in the model having to do with peer relationships, and that we think that if we can improve these functions, that they will develop more normal peer relationships that are healthier and not based on conflict and some of these other things. And that in turn is another mediator of the effects, will lead to better behavioral outcomes. And so so we haven't done it yet, but I just wanted to let you know how other models can be broken down like this. The biggest thing here is the reward sensitivity. Reward sensitivity it's been very well charted in terms of its neuroanatomical, its functional neuroanatomy in the brain. Okay, so we know a lot about reward sensitivity. It seems to be a steady state, as a steady trait, as opposed to a state. So we don't expect to be able to move the needle on reward sensitivity like we would for error monitoring and emotional regulation and so forth. So we see it as a moderator. And interestingly, with GBG, it works best on aggressive boys. And the literature, if you look at the conduct disorder literature and the delinquency literature, even from criminology, that these are the kids that have the highest levels of reward sensitivity. And so we can actually sort of capitalize on something that can be a liability and use it to alter behavior in an advantageous way. See what I mean? Well, anyway, I'm excited about that.
When you say steady trait, does that mean that this person has sort of a level?
Yeah.
Pretty much their
Pretty
entire
much,
life.
yeah.
And
Like
some other person
temperament.
may be higher.
Yeah.
But I see.
Yeah,
Okay.
yeah, like temperament is
Mhm.
relatively stable throughout life unless something traumatic happens.
Hmm.
And so this slide just shows how, what we already know, that the accumbens shows the reward sensitivity that is enhanced in adolescents compared to children and adults. And so that's just, a developmental phenomena, normative, but it shows that in general adolescents have more reward sensitivity and that it's been fairly well charted out in the brain. So and again, to maximize on these things that we know about the brain. So the overall goal is to strengthen vertical control, top down. Effective programs theoretically capitalize on the brain's plasticity. This is what we need to know more about. This is the program of research that we think is at the forefront, one of the many areas in prevention science that need to be further delved into. And that this one actually refers a lot to mindfulness, this last bullet about intervention enhanced vertical control, where we're looking at awareness and attention to regulation of emotions and somatic sensations. So that really has to do with mindfulness, but there are other programs that work on that, such as PATHs. And then there are other programs that don't. So there are other ways of enhancing vertical control that we think will be measurable. And so these measures are, considered to be much more sensitive and specific than the measures that we've used in the past in prevention science, which are largely surveys and so forth. So I should probably stop. I think there's only 10 minutes left. And I was going to go into optimization, but all of this will lead to optimizing so we can identify what the active ingredients are, the components that have the biggest effect on neuroplasticity that will lead to improvements in our clinical outcomes. And so the ultimate goal is that through a transfer of knowledge from etiology to practice and back to etiology, clinical and public health policies will be increasingly responsive, applicable, and precision-based, thereby exerting greater impacts. That sort of describes what my ultimate goal is in doing this. And to facilitate the transfer of that knowledge, I run a national coalition. So these are the sorts of things that we do. We have scientists from all these sorts of disciplines. We have educators and clinicians and practitioners and community organizations and advocates and policymakers. There's about 550 of us now and growing. And we actively transfer what we do know. And of course, we all still have jobs as researchers because there's a lot we don't know. But we actively transfer what we do know so that we can make an impact with the interventions that we have that will have beneficial effects. And to make sure that we have relationships with policymakers who are integral in making policies that are frequently not, are not conducive to optimizing the human condition. That came out funny, but you know what I mean? Yeah, I'm trying to be politically decent.
Or perhaps not.
Wasn't exactly politically correct, but it was trying to be decent. But yeah, so, and we do congressional briefings on a regular basis, like every 2 months, about what the evidence base is and what we can do. And our one next week is different. It's on policing. It's on police community relations and the violence that we're seeing in communities, but we're bringing prevention science into it with respect to new strategies that have not been tried, for this kind of issue. And we're also bringing to the fore the fact that, that mental illness is often unrecognized by police officers and how to deal with it. And the PTSD and trauma situations of kids that live in the neighborhoods in Chicago and Baltimore and so forth is frequently not recognized and dealt with very well by police officers. Inversely, we also do not recognize the PTSD that often exists in police officers. And so, let's talk about emotional regulation and try to remember that the ultimate goal is public safety. And that's the goal of the community and the police. And because of the conflicts that we have, we frequently forget that. So, so that's just the next one. And then the one after that, we're going to talk about the economic benefits of prevention. We've done one on poverty. It's all on the website. You can sign up. It's free. You don't have to pay to be a member, and you get occasional updates about what we're doing. So I'll stop and see if there are any questions. Can you all hear me in Hershey?
We can, yes.
They were gone from me. I have to do it again.
Sarah, are there questions down there?
Nope, I think we're good here.
Do you, as part of characterizing, let's say, these subpopulations, are you looking at actual biological markers, cortisol, or some other things?
Yeah.
Sort of a battery?
Yeah.
Yeah.
We use the markers that we think are going to be involved. So stress physiology is pretty, standard because of the populations that we look at. But, the performance testing that we do depends on how the components are mapping to these neurocognitive prerequisites. Like I showed, for the PAS study, the ones that we had are different from the ones that we would use for GBG, which are different from the ones we'd use for mindfulness. But we do the neurocognitive testing. We have a stressor task, the different tasks that we use for different developmental ages. We don't want to upset anybody, so we use subtle stressors. For little kids, we use a story stem battery with, little playground conflicts and somebody took the ball from me type thing. And, and then they finish the story and we measure their physiology while they're doing that to see how upset they are. And then we can see change over time because, they're all intervention studies. So we have to look over time to see if their stress physiology is starting to normalize. And then some studies we use imaging. If they're too young, we do not. And if we don't have enough money, we don't. But yeah, or if it's not needed, sometimes you can do these things. I've got one also designed that is going to go into review for multi systemic therapy, MST, for juvenile offenders. And I have a different model for that altogether, where it's they focus on family and the school environment and the community and service providers and so on for kids who are in trouble with the law. And I don't think we're going to get pre-post intervention effects. I think my new model is that behavioral change is going to drive change in the brain. Like generally you can see in my models are mostly about the brain driving change, right? So the intervention acts on the brain to drive change in behavior. But in this model, the intervention drives change in behavior that over time, with practice and sustainment of behavioral change, a new behavioral repertoire, right? Where you're practicing inhibitory control and so forth a year after the intervention, that the kids who are still sustaining that change are the ones that are going to start to show brain change. And it takes a while if you don't have a really intensive, treatment of some sort. So, and then we'll be able to see the difference between those that improve over time and those that don't. These guys that don't improve are less likely to show any change in the brain. Well, normal developmental change. But these guys should show an acceleration of neurodevelopment that corresponds with their behavior.
Interesting.
Yeah.
That was great. Do we know how long the changes in the brain from an intervention might be sustained?
We don't. We theorize that they would be longer lasting than some of the things that we're just measuring in surveys, attitudes and things like that. intent to change.
Mhm.
A lot of, surveys are measuring the intent to change, or how would you behave in this scenario, and so, yeah. They're very indirect measures.
Mhm.
And we have to use them. It's not like we don't use any surveys. We
Yeah.
still have to do that. But we think that if you're actually acting on the brain's plasticity and connectivity, that you're likely to have sustained change.
Mhm.
But certainly there also has to be sustained change in behavior and no trauma,
Mhm.
right? Another,
Mhm.
severe incident of trauma is going to derail
Mhm.
that trajectory. Anything else?
Thank you very much.
Oh,
Very
good.
interesting.
Good.
And I hope lots of you will think about registering for the symposium next
P-TRAN,
week.
yeah.
There'll be lots of
P-TRAN is this whole
discussion.
program of research,
Yes,
yeah.
right.
And we brought in a lot of people from across University Park and Hershey to be part of P-TRAN. And so it's on translational research on adversity and neurodevelopment. And so we have the 4 areas of interest: mental health, and I guess you already know. So adversity and stress, child development. And so our various people have been asked to talk about their work and at the end bring it into a translational context. What would you do with this basic science to translate it to the next phase being program development? How would you, potentially design a program based on your basic findings? Because that's what translation is. You got to move to the next step.
That's right.
Yeah.
Yep.
So come.
Great. Thank you
Yeah.
so much. We really
Oh,
appreciate
well, thank you for inviting
the
me.
opportunity to
Totally.
be able to hear about this.
So
Sarah,
Sarah,
we're good?
did anybody else show up, or you still have 3 people?
Oh yeah, we're holding strong with 3.
Oh, good. Okay, 3 strong people. Thank you for your
Thank you,
patience
that we
over there.
appreciate it.
All righty.
Thank you.
Take care now.
This is such a tough time of year.
It is. It's
Um,
probably the worst date because school just ended at graduation.
People are running and